Hello everyone and welcome to Cure Talks. I'm Shweta Mishra and today we are discussing the nuances of COVID-19 vaccines in multiple myeloma patients with Dr. Zainab Shahid, who was a member of the team that helped develop the COVID-19 guidelines. Dr. Shahid is an infectious disease specialist, medical director of bone marrow transplant infectious diseases at Levine Cancer Institute, and clinical associate professor of medicine at University of North Carolina, Chapel Hill. Joining us on the panel are myeloma patient experts Gary Peterson, Jack Aiello, and Cynthia Schmielewski. It's my pleasure to welcome you all to Cure Talks. Dr. Shahid, Gary, Jack, and Cindy, thank you for being here on the panel today. We will be taking in questions at the end of the discussion based on the availability of time. So please send in your questions on the talk page on curetalks.com or on the YouTube chat window, or you can also email them to shweta@trilex.com. To begin with the discussion, Dr. Shahid, it would be great if you could begin by giving us a very brief overview of the technology used and the dosage regimen of the available and soon to be available COVID-19 vaccines.
Certainly, Shweta. Good evening, good afternoon, good morning, everybody. It's a pleasure to be here invited with such esteemed panelists. I'll start just very basic three mechanisms which are currently very close to being approved or are already approved for the mechanism of action for the vaccine which are currently available. We all know about the messenger RNA technique where what is done is that a messenger RNA is injected into the body. It is like a code that goes into the host, which is the recipient of the vaccine, and generate proteins using the host machinery to generate protein just like the virus. Currently, the protein that has been utilized for this decoding is the spike protein of the coronavirus. And so the Moderna and the Pfizer vaccines are the two vaccines which are already approved under emergency use authorization in the US and being administered. That's one mechanism of action. We know that they're safe. Has been used this technique has been investigated actually for decades. There are other vaccines such as the CMV vaccine that has been being designed and being studied. So it's been studied, this technique. However, we were lucky that this was utilized for the SARS-CoV-2 virus, and it is pretty safe. The other two important mechanisms which are very close to being approved would be the adenovirus vector vaccine. What happens is that you have a vehicle, which is the adenovirus, that takes the gene coding of the part of a protein of the virus. That combination goes inside the human body, and then the vector, which carries the code, is taken inside the cells. The vector allows entry into the cells, and then the gene that have particular protein is coded by the human body, and then the immunity is generated. So AstraZeneca is the vaccine which has been used for this, and then also the Johnson & Johnson, which hopefully will be approved by the end of this week, is made with the vector technology. And lastly would be the what we called a protein subunit, meaning you have a protein and then you also have a protein of the virus and something that is attached to the virus protein and is given to the human body, and then they generate, when it is identified, the human body generates immunity against that protein. So these are the major three mechanisms which are being utilized amongst others.
Right. Thank you so much for that comprehensive overview, Doctor. And I have read that the timing of vaccination is of great importance while planning to vaccinate myeloma patients. So we have gotten quite a few questions from the audience that are asking about the timing of vaccination. So I'll read a couple of them for you to so that you can answer it collectively. So the first one says, should maintenance therapy of bi-weekly Velcade injections be withheld before or after 2 vaccine injections? I have seen information suggesting not to have maintenance therapy until after the second vaccine is received. please advise. And the second one talks on the same lines about Revlimid maintenance therapy. So if you can answer those.
Certainly. So this has obviously been a very important discussion because all myeloma patients are very much concerned if they do receive the vaccine, would there be benefit? We do, and what would be the best time to do so? I can tell you that no myeloma patient is the same. They're all at the different stages of their treatment. There are at different durations in the treatment. A lot of timing of the vaccine is also dictated by states when it becomes available. So there has been no, would say, consensus because certain people would say it's better to hold, but can we really do that given what the timing of the vaccine would become available? Because that's not dictated by many medical centers. It's dictated by the state. You have to get the appointments. So there is no right answer to this. If we are able to hold, and if there is a discussion between the patient and the physician that it is safe to hold therapy, I think we can. And if it is believed that we just can't go around the cycle, it is important to continue chemotherapy. And it is also important to get the vaccine. I think that we should proceed while they're on maintenance. So there's no right or wrong. We do know that the current vaccines, the messenger RNA vaccines, are very good vaccines. They're very immunogenic, meaning that they would produce a good response. And we hope that they can do so in our immunocompromised multiple myeloma patients as well. So I can't give you one clear answer. It has to be the discussion between the patient and the physician, but either approaches are fine. And for the Revlimid, like the 21-day cycle patients, it is also recommended, if possible, you can give the vaccine in the off week. However, it may be possible each time. So should we not get the vaccine? I think it is okay to get the vaccine at any point at that time. So it's going to be a very tailored, individualized approach, knowing that overall, at any time the vaccine can be given, better to be not on chemotherapy, but if you are, it's fine. I hope that helps.
Absolutely, yeah. So it has to be an individualized approach. That's what you said, Doctor, yeah. So I will read out the next question, which is again on the same lines, timing of vaccination, and it relates to stem cell harvesting. So the person says, I'm scheduled to have my first COVID vaccine on Feb 28th and my second on March 20th. And I'm having my stem cells harvested for freezing on March 11, 12, and 13th. I'll be taking Zarzio and Mozobil on March 10, 11, and 12. And my doctor thinks that it is okay to take the vaccine. Should I be concerned? And he or she also says that I've been off chemotherapy since January 27th, and I remain to do so until after the harvest. So if I do everything as planned, how soon can I start treatment again?
So there isn't a contraindication or, some in this clinical particular scenario, in my opinion, there's nothing holding or the patient should be able to get the vaccine. There is no reason not to get the vaccine. Now, again, I would say treatments are so individualized. Maybe it is important for that patient to be on treatment. Then that patient should have a discussion with their physician how important it is to continue chemotherapy after the harvest. So it is I do not know the stage of their multiple myeloma, what risk factor what do they have, whether it's a high risk myeloma or low risk myeloma, how important it is to receive chemo before they get to transplant. So there are so many unknown scenarios, but as an overall, it is okay to receive the vaccine with the timelines that is described here. And the harvest shouldn't be interfering with the vaccine or vice versa.
Sure. Thank you, Doctor. With that, I will hand it over to Gary. Gary, the floor is all yours. Go ahead.
Okay, well, thank you very much, Shweta. And Doctor, thank you so much for talking about one of the most troubling things that are confronting myeloma patients to date. And because we know, based on some information that I've seen, that we're probably one of the very highest risk groups, meaning about if we get COVID, we have about a 33% chance of not living through the episode. So, obviously things like the vaccine are very, very important to us. Now, you talked about whether we should or shouldn't get the vaccine, and it's pretty much that you, along with many other doctors, including Dr. Fossey, that we should get because any any immunity is better than none at all, which is kind of good. But given that, is there any evidence that myeloma patients can obtain immunity? And that's in, during treatment, after treatment, several years after treatment. Is there any evidence? And I'll preface it by saying that I know that, and I mentioned it to you before, that Johns Hopkins has just started a study where they're testing after the first shot, after the second shot, and then later to see whether or not there are robust antibodies developed in those patients. So I ask you, is there any evidence? Have you witnessed a robust response from the vaccine to date?
So the evidence for specifically for multiple myeloma patients is being generated, just as you mentioned, the Hopkins study, and there are other institutions which are developing protocols to test antibodies after the vaccine. We know that the antibody data that we currently have exist only in mostly healthy individuals of different ages, based on the studies that got this EUA approval. So it is important for us to understand that we are generating evidence as we are receiving the vaccine. I cannot go to a single study to this date that would say this is how a multiple myeloma responds. So there are two important things. As we measure levels in these patients as they get the vaccine, it will be also very important to interpret what that level means. At what level of immunity do we have really protection from the infection? So there is much to be known. These are exciting times. What I can tell you, what I really recommend, even after the vaccine, make sure hand hygiene, social distancing, and face masking. Very important, because we do not know for our very vulnerable multiple myeloma immunocompromised patient what immunity means, what vaccine efficacy means. So while we are big advocates of vaccine and immunocompromised patient, at the same time, protection with these measures is also very important.
Well, thank you, doctor. And the other thing that I think is important to us is in the case of the flu virus, for example, they found out that older adults need a mega shot, I guess, or something that has far more, a bigger dose, or I don't know exactly what the thought is there. But, they decided that because for immunocompromised people, at least with respect to age, that they have to modify the delivery and the dosage for flu. Do you see the same kind of thing might be required for COVID-19?
It's a very good question, because as we get to know more about the responses in immunocompromised patients, and also we also have to take into account the fact that there could be some mutations within the virus that may require the virus components be changed in the near future. We do not know. So I do believe that there is going to be some form of revaccination, as we know when the immunity actually wane off. So we would never know from the clinical trial patients, only because everybody who was participating in the clinical trial on the placebo arm actually got the vaccine afterwards. So it would be checking levels thereafter and collecting real world data, which is of so important to us, so I better understand how soon is the reimmunization would be required. Is it every year, every 2 years, every 6 months? Do we need boosters? Or do we need to change the ingredients of the vaccines, just like influenza?
Well, thanks again. So, given that, I think that what I've read, if we're looking at somehow solving this issue, it seems like what we, if you 500,000 people have now died from this, which is more people that somebody said that have died in the military action in World War I, World War II, and the Korean conflict, which is it's beyond belief. And the question is how do you stop the deaths, right? That would be the important thing. I mean, that's probably the most important thing. You want people to quit dying. And in the UK, they have a nationalized health system. And then in that system, they've now determined that people who die from COVID-19, 96% of those people happen to have a pre-existing condition. And given that only then 4% or 5% are of the patients which don't have a pre-existing condition die, that it looks like the target population, if you want to eliminate 95% of all deaths, is that particular group of people. Now, I know that Florida, for example, is at least allowing people 65 or older, but only probably 50% or so of that group has a pre-existing condition. So it seems like in some cases we might be, overkilling that population. On the other hand, those that are have pre-existing conditions that might be 50 might be far more likely to die. So I guess my question then is, if this is so important and we can get control of this virus by stopping the deaths, and the deaths are with people with pre-existing conditions, I'd say why the HE double hockey sticks aren't we doing that like they are? They happen to be doing that only in one place, and that's in San Diego, where they are looking at people over 65 with pre-existing conditions. So that would be my question. Sorry for the long.
No, it's a very important question to be asked, and it also is of high impact, meaning we can achieve more by targeting the most vulnerable. So in preventing. So this speaks to two things. Number one is decreasing the spread in the community, because everybody lives in the community. Number two is vaccinating the people, number one, who spread the virus, number two, who have adverse outcomes from the virus. So I think that so far the hurdle has been that each state has had their own way of distributing the vaccine. For example, in the state of North Carolina, we already vaccinated 65 and above. We started with 75 and above, and healthcare workers. So this has not been it has been a different statewide approach, and the distribution of the vaccine has been handled by the state. And we are, for example, as a healthcare system, are following the mandate. So it would be, if we can just vaccinate quicker, the more people with comorbid conditions, I think it would be a very high impact in preventing. The only thing is that if we don't have different states have done it differently. And I think it depends on the supply, depends on the resources that they invest in trying to vaccinate, because vaccination programs also require resources. And then also what's interesting, what we have found in our institution is the uptick. So no matter how much you want to offer the vaccine, the uptake started only at 30% and then went up to 40%, then up to 50%. So it's been a complex, I would say, problem. It's not as simple as the vaccine we saw offered was an uptick by people as well, because of some of the misinformation out there. So I hope there's hope. I think we're closer. I do believe that more and more people are vaccinating. platforms such as these, we are speaking out more, educating more, I think it will get better for sure. Yes, but I do believe that 500,000 is a staggering number.
I asked that question. I've got a call from one of the people that follow my website, and he says, my wife is stage 4, multiple myeloma under treatment, and she's like 65 years old, and he can't get her vaccinated, because it's 75 and older, and he's tried, and he can't get it done. And he was asking me what to do, and I says, I understand. I'd be going crazy, but
And I think in this scenario, looking at the state, wherever state they live in, their website would be the most information that they would be able to get. And we have sometimes reached out to the state representative. We've done that, where we are trying to get vaccination for our household members. The best way to protect an immunocompromised patient is also to build immunity ring around them with the household members being vaccinated. So speaking up, reaching out, trying to speak to your representative would be another way that they can advocate for the person to be vaccinated.
Biden doesn't answer my emails.
I do believe that the state health departments would be the best. That's what we are approaching. We're reaching out to the local state health departments.
Okay, thank you so much, Doctor. I think it's Jack. Are you up?
Yep.
Or is it Cindy? Okay,
I'm up.
good.
Dr. Shahid, first of all, it's very nice meeting you virtually, and I appreciate your taking the time to be part of this conversation. When you began, you talked about the different mechanisms of actions. And while I don't understand them completely, I keep hearing the Moderna and Pfizer vaccines, which have the same mechanism of action, don't prevent one from getting the disease, but rather reduce the impact, the effects of the disease. First of all, I want to clarify, is that true? And then secondly, are the others mechanisms of action meant to eliminate or reduce the ability to get the disease?
So definitely, it's a very good question. And what I want to, so there is one number is, does it decrease the acquisition of virus? Number 2 is, does it decrease the acquisition of symptomatic illness? So the studies that were designed were not designed to study the acquisition of virus, meaning people did not get tested till they have the symptoms. So what it means is that we don't have data to say that it decreases the acquisition of virus. We just don't know.
Yeah.
And so it does, all the studies have been geared in the pandemic setting to look at the morbidity or mortality as opposed to the acquisition of virus and subsequent morbidity and mortality. I do believe that there is going to be studies designed. There were some published data from Moderna that it did decrease the acquisition, but obviously that study is not, those studies weren't powered or designed to study it. So I think there would be more information in the future. We just can't say anything without the presence of evidence.
Got it. And do you expect the other trials for the other vaccines to show anything different in terms of acquisition?
The trials that I know that currently are phase 2 or in phase 3 are close to aren't designed, but obviously I haven't looked at each individual protocol. I know that Johnson & Johnson and then even Novavax, which are currently being done, well, Johnson & Johnson is close to approval, Novavax is being done. They're only testing when symptoms occur. So they're not doing swabs on asymptomatic patients. So we would never know the acquisition of infection was decreased because there's so many resources and it requires so much money. And then there is this pressure to look at the high impact morbidity and mortality. But I do believe that there will be studies in the near future because obviously with this data, this huge question came up because that would take us away from not wearing a mask and not getting all this virus within the community, but I do believe we will know in the future, but not currently what is being presented in the near future.
Thank you. And in your answer to one of the questions that came in from the listeners, I think you answered one of my questions almost completely in terms of dosage adjustments or when you should get the vaccine, except as it relates to transplant. I have heard that if you get a transplant, you should wait a couple months afterwards before getting a vaccine. Can you confirm or deny that?
Sure. So we looked at the data extensively and produced the American Society of Transplant and Cellular Therapy and American Society of Hematology guideline. We recommend receiving the COVID vaccine 3 months after the transplant. And that would be the earliest that one should receive, because at that time we believe that the immune system has recovered enough for it to mount a good immune response to the vaccine. And so those are the guidelines now. Now there will be studies in the near future. We are discussing different platforms where you can actually study the immune response post-transplant. So there will be more data forthcoming in a year or so, or in 6 months or so, as it gets as people receive vaccines.
And my last question has to do with the various institutes like Johns Hopkins and I know UCSF, where I am, are doing their studies of antibodies after receiving the vaccine for myeloma patients. One, are they sharing data with each other? And two, does this study include, and I suspect it does, information on the treatment the patient was on during the time of the vaccine?
I will answer the second part of the question. I do believe that when the databases are created, they are created in context of the treatment stages, the multiple myeloma stage and treatment that is being done. And I can tell you what one thing we have learned with COVID-19, especially on cancer patients, is that sharing data is the biggest power that we could use so that we can benefit many. So I cannot imagine anybody not sharing their data as it becomes available, because I think we can tweak the timing. Then we can also tweak to see if they need more doses. So there is a lot that will be understood by these studies, and I'm sure that the institutions will be sharing these data.
Thank you very much. I appreciate it. I'll turn it over to Cindy.
Hi, Dr. Shahid. Welcome. And I guess we talked way back in the beginning of this virus when we were just wondering how we should protect ourselves. There was no vaccine. So it's nice to now talk about the vaccine. And my first question was, some patients are concerned that, after they get the vaccine, they really aren't experiencing any side effects. They're not getting a headache or anything but a sore arm. Does that mean that the vaccine's not working? Do side effects equal response?
When you look at the trials that are presented and look at the incidence of side effects, they're not 100%, but we do know that they're anywhere between 50 to 60, depending on what side effects that we are looking at. And we know that, for example, efficacy is more than 90% in both the vaccines, up to 95 with Pfizer. So the presence of adverse events or side effects after the vaccine does not translate into immunity or your body immune response. So you're right. I got this question so many times already, and there's this conception that it means that you have to have side effects, which is probably not true.
Okay, well that's good to hear, since I didn't have many side effects. So. And the next question about the vaccine is, does the COVID vaccine, or could the COVID vaccine impact any of your myeloma labs? For example, if you got the vaccine and right after had your light chains drawn, could they be elevated unusually just because the virus is working? And also in that respect, could you talk about the vaccine and women? I think I read that women should not be getting mammograms right around the administration of the vaccine because it may involve some of the lymph nodes. So any insight you could give us on those areas?
Happy to, and it's a very good question because we do come across in our inpatient setting with this scenario. I do believe that after the COVID vaccine, one may, and there is data lacking, may experience a increase in the inflammation in the body. Not everybody, maybe a selected few people who have an inflammatory response. And one of the side effects is actually injection site irritation and some irritation of the lymph nodes on that side of the body. Outside of those rare scenarios, I do not believe that the specific multiple myeloma labs will be affected. And I also believe that when you're looking at mammograms look at obviously the lymph nodes, but the basic thing that they look at is breast tissue. I do not believe there would be impact on the breast tissue outside if there's somebody has experienced lymphadenopathy that might show up and could be misread or misrepresented. But as a rule, I do not suspect that it would interfere in either of the testing.
Okay, good to know that too. Now, you might have answered this question when you were speaking to Jack, but I wasn't quite getting everything you were saying, so I'm going to ask it again. Could you explain what like viral load is, and can it be measured, and how is it measured? And if someone was diagnosed with COVID, if they had a lower viral load, does it mean they're less sick? And then would they not spread the disease as quickly? And finally, then would having a COVID vaccine lower your viral load if you got the virus? So anything about viral load to make me understand it a little bit better.
Definitely, it's a great question, and I believe that there is not a lot for me to tell you, only because we don't have any FDA approved test that actually looks at viral load for SARS-CoV-2. Viral load means the amount of virus in any given specimen in a given amount of specimen. For example, per ml, per amount, how much virus is replicating in a given tissue. We do know that the SARS-CoV-2 can be present obviously in the respiratory tract, but from sicker patients inside, and when the studies were performed in academic settings, we know that the virus can be present in the heart, the brain, the lung, every tissue. And there are ways to quantify the virus, which is called the viral load, but there are no standards for it. What does a high viral load mean versus a low viral load mean? We do know that the amount of virus can be present in asymptomatic individuals, similar to the symptomatic individuals. These are studies which are done looking at the respiratory samples. What it means, do we believe that the viral load would be higher in a sicker and hospitalized patient? We do believe, but we haven't proven that yet because there are no standard testing that is available in the outpatient setting or in the inpatient setting. So we are learning much more what we called the kinetics of the virus, how it gets acquired, how much does it replicate when we actually get rid of it. There are some data around shedding, meaning just the presence of virus in the DNA. There's more data around how long we can shed the virus, but what is the quantity of the virus? It is still being studied and there's nothing standard. So we would believe, based on what has been published, people with the low viral load may be less infectious, but it may not translate into sickness. I wish it is one of the dreams for COVID that I can actually test the people so we can cater antiviral treatment based on the amount of virus, but we're not quite there yet.
Okay. There were my questions, but I do have another if there's no other questions.
Yeah, go ahead, Cindy.
Okay. don't get mad at me, Jack. He's like laughing, like she always has questions. It's with the antibodies. Now, does the amount of obviously if you make more antibodies you have more protection, but does everybody need to make the same amount of antibodies to make the protection? Like, can I make 10 and be protected, but then Jack needs to make 50 to be protected? Or is there like a certain threshold that you need to be protected? Does that question make sense?
It's a wonderful question, and it is being studied. So let's put it that way: there is immunity, or let's say development of antibody, because we do not know what amount of antibody means immunity. That is, we haven't developed what we call threshold or cutoff points. That after 10 means protection, below 10 does not mean protection. So I just want to make sure that we understand that there are no thresholds being developed. However, we believe, based on the data, that one does develop some protection after natural infection because of the rates of reinfection within 3 months of original infections were low in patient populations. So their immunity does develop. What antibodies? So there is, once patient develop antibodies, there are multiple different kinds of antibodies that develop. So which particular antibody to study? Anti-spike protein, neutralization antibody. So there are so many antibodies to be studied. So we do not know where we would draw the line of the threshold, but I do believe as we are developing studies, especially after the vaccine, we would reach a level, we would know that after this amount of antibodies that are being developed, there would need protection from reinfection. So what great points, but we're just getting there. We're not there quite yet.
And well, if I make antibodies, but because I have myeloma and the way my myeloma makes antibodies is sometimes messed up, would the antibodies, even that I make, they get measured that I have antibodies, be functioning antibodies, or don't we know that?
We are hoping to know that. We don't know that yet, because there isn't studies that are done where we have looked at the level of the antibody, and then, for example, somebody's IgG levels, whether they were good and somebody got the infection, so did they really function? And that would also be true for specific for COVID-19 antibodies. So there's lots to be learned, and especially in our immunocompromised or patients on chemotherapy, there is still much more to be learned for this. And there is a lot of interest in testing antibodies because we want to know more, especially in our immunocompromised patients. So we hope to study that and follow patients clinically at what level did they actually got the infection. So there's much more, there's exciting time. The good news is that we have the vaccine, and the vaccine we believe works very well. And so we hope that because it works so well, it will work well in our immunocompromised patients as well.
One question, one question I have as well is I've gotten both shots now. And as a myeloma patient, see, we know that, we can get a cold. Like for me, I can get a cold, and before I know it, I'm in the hospital with sepsis, because we just don't have the immunities. But now I've got this, these 2 shots, and I'm getting a little cocky, actually, as far as, I may go out to a restaurant. We did go out to a restaurant. We social distance and that kind of stuff. Now, is there an issue with that? Or can we do more things now that we've got the shots?
So,
and to preface that, have you gotten your shots?
I got both my vaccines.
I hope so, because you got a bunch of COVID sneaking up on you there in the background.
Yes, I did get my vaccine, but I still recommend using caution, only because I do not know what the vaccine. Well, I do believe that the vaccine will really decrease the severity of illness in any given patient. If they didn't get the vaccine, I think anybody to develop illness would be severe. I have complete believe in that based on what I have studied. However, I would still recommend all the social distancing. I still am hesitant to recommend everybody to eat out and be more free, because when I see the level of community spread, I do in our state, at least, I'm telling. So it has to be in context of what's in this community. If most of the people in the community are vaccinated, or the level of spread is less than 5%, for example, it's a different precaution or ease that I might have, say, having somebody it's okay to eat out, compared to if somebody's having a community presence of 15% or 10%. And then it means it's still developing the community, it's still being spread. And so what would it mean after, what immunity means for my vaccinated immunocompromised patients, I am still learning. So I believe that it has to be multifaceted rather than one person got vaccine, now they're protected. But I do believe the people around them and the community around them also provide a big safety net for immunocompromised patients.
Well, thank you,
Okay,
Gary. Thank you, Cindy. Thank you, Jack. Before we move on to the audience questions, in the interest of time, we do have Dana Holmes, smoldering myeloma patient advocate, joining us, who has joined us via telephone, and she has some questions for Dr. Shahid. So, Dana, if you could unmute yourself and ask your questions. Dana, you there? I think she just dropped out. She was here waiting for us. So I guess I'll just move on with the questions that are posted on the page, and maybe she'll join back. So Dr. Shahid, there's one question that was posted on the page, and it asks, is it okay to take non-steroidal anti-inflammatory drugs for pain or fever after getting the vaccination?
There's no data that they would interfere with the production of immune response. We generally do not recommend our patients to do so as a routine unless they become debilitated and they're unable to do what they need to do. So I believe that it's okay if the symptoms require attention. Some people would scared, oh, I'm going for the vaccine, I'm going to pop in a few Motrins. That we do not recommend. But we don't know any data that you cannot take any of the non-steroidals.
Okay. I think we have Dana back. Dana, could you please unmute yourself, and if you'd like to ask a question?
Hi, Priya. I'm sorry. I'm so technically challenged. I'm hoping that you guys can hear me.
Yes, we can hear you completely. Yep.
Oh, goodness. Thank you so much. It's Dana. Hi, Dr. Shahid. How are you today? It's so nice to chat with you and listen in on your discussion. Learning so much. Piggybacking on both Jack's and Gary's question. I appreciate you finally fine-tuning the messaging regarding the impact a vaccine would have to actually prevent infection. You shared that we don't really have evidence of that yet. So we would need to expect this in the myeloma patients who are vaccinated as well. So even with a potential blunted response to a vaccine due to the very inherent nature of our cancer, would we at least expect to not progress to a severe disease course of COVID if we did become exposed and infected to it? Is that the hope, at least, the minimal hope for the myeloma community?
Oh, very much so. I have true belief, based on what has been the studies show, I do believe that it will provide protection. I just can't tell what would be the level of protection. So for example, if we did the study solely on multiple myeloma patient, would the efficacy be 95%? I think it would be less than that, but I do believe that individual people would get benefit, even with the attenuated response to the vaccine.
Great. That's very hopeful for us, because, you know, we, just like everyone else, we've been holed up in our homes, and, you know, Gary's luckily out and about now with his double dose. Be careful, Gary, please mask up.
Yeah,
And
apparently.
yeah, regarding dexamethasone, obviously patients receive last-minute notice about coming in for a vaccine. So if they're unable to modify any of the other meds, Revlimid, Velcade, anything else, would Dex be at least an important one not to take on the day of the vaccine or the day after or any other additional time after that if they potentially could avoid taking it?
Yes, so we hope that there is time to allow a discussion with the patient, but we do believe that we could hold DEX if possible. You know, in any, it's individualized, like I said, I can't comment as a whole, but yes, even in our institution, if we can hold DEX that day of or the next day, we are holding it. And I think the International Myeloma Society guidelines also recommend that. And when we look at the NCCN and all of the guidelines, you know, basic focus is to just get the vaccine, no matter what. That's the bottom line.
Right.
It would be
Right.
great to have the opportunity to hold things and to modify things, but most of our times than not, we won't be able to. The message is just get the vaccine wherever you are, in what stage you are, no matter what chemo you're receiving, and talk to your doctor. So yes, if possible, let's hold DEX, but it's not absolutely 100% necessary.
Okay, great. But what would the reasoning be to hold a drug like dexamethasone? Is it known to dampen the immune response somewhat to a vaccine?
So we have not studied the messenger RNA vaccine and its steroid effects on the mRNA vaccine responses. Well, but we know that dexamethasone and it's a very important modulator, immunosuppressant, and anti-inflammatory. So there is hypothetical risk that it would attenuate or dampen the response. But we do not know that for sure. And I could be completely wrong because we haven't studied in particularly with the mRNA vaccine. But the thought is that if you can, for example, for our graft versus host patients, we don't immunize patients when they're receiving steroids because of the same reason, because we believe that the response would be attenuated.
Oh, great. Thank you for that explanation. And thank you so much for your engagement in our online COVID Facebook group. It really has been very, very helpful, along with the other myeloma specialists who signed on to our group. Appreciate it very much. And thanks, Priya, so much for the time.
Thank you. Thank you, Dana.
Dana, and thank you so much for rattling my cage as well as Priya Menon's in order to have this specific Cure Talks on COVID-19. So thank you for doing that.
You bet, Gary. You bet.
Yeah, so I'll just continue with the audience questions that have been posted on the page, quite a few. In the interest of time, I'll just, I think the antibody question Cindy just asked, thank you for asking that, Cindy. There was a question that somebody wanted an answer for that. So the other one is asking, how long after getting the second vaccination would it take if one wants to find out how much of an antibody load was generated? I think that part was left in your answer, doctor.
So usually 30 days, not before 30 days, we would be like for you to get the antibody response testing done if somebody is checking. Different institutions may do different protocols to look at what would be the response like for 2 weeks or 30 days or 60, but outside of those protocols, looking at a true response would be 30 days after, at least 30 days after the first vaccine, maybe 60, depending on where a patient is and when you can get it done.
Okay. All right. So I'm reading out the next question, which says, should a multiple myeloma patient in remission but on treatment with darapomelis dex, for instance, try to get one of the RNA-based vaccines with higher efficacy rate, since it's unknown how well our immune system will respond, or if the patient is offered Johnson & Johnson vaccine, they should take that?
So the sooner you get it, whatever you've had hands on, just get the vaccine. It doesn't matter which one would it be. but I wouldn't say miss the second dose for sure. So if you're getting the Pfizer, please make sure that everybody understands we need two doses. Even though there are data been published that even one dose is good, just make sure we need to understand that we need to form memory cells. We need to have a robust immune response, and we know that the 2 doses will do that. So making sure that whatever dose recommendation are for that vaccine, just get that vaccine. And whatever you get to, just get to it.
All right, thank you. Yeah, you mentioned memory T cells. So we have a question asking, are we developing tests for memory T cells responses to the vaccine?
There are actually development assays which are developing, looking at specific T cell responses for SARS-CoV-2. Obviously, nothing is FDA approved. This is all in context of research. But just like antibody, there are assays that are being developed to look at specific T cell responses.
All right, thank you so much, Doctor. I think we have covered most of the questions that were posted. And thank you so much for all the information that you shared with us today. And I'm sure it will be helpful to the myeloma patient community and their caregivers. Gary, Jack, and Cindy, thanks for joining the panel today. And this talk and transcript will be available on curetalks.com for everyone to read. Stay tuned for more upcoming talks on curetalks.com. Until next time, thank you, everybody, and have a great day. Stay safe.
Thank you, Shweta.
Thank you.
Thank you. It's a
everyone.
pleasure.
Thank you, doctor, very much.
Thank you, everybody.
Care.